Hello everyone, welcome again. Today my video topic is regulatory RNA. So before starting the video on regulatory RNA, I want to tell you about the basic structure, basic definition of regulatory RNA. So as we all know that there is a central dogma process, that is from DNA. mRNA is formed with the help of transcription process, and from the mRNA proteins are formed with the help of translation process. So this follows the central dogma process. And in the regulatory RNA, basically as the name indicate, regulatory RNA, so means there is a piece of RNA or mRNA which can like regulate the expression of the same gene. It can enhance the expression of any gene, or it can inhibit the expression of the same gene. So basically regulatory RNA act as a regulator which can switch on or switch off the expression of the gene with the help of ribosome binding site or ribosome site. Means here regulatory RNA is a part of RNA which use the site of ribosome binding site. As you all know that during transcription we have a product, that is mRNA from the DNA, right? And from the translation we got another product, that is protein can be a regulatory protein. So basically what happened in the mRNA structure, in mRNA structure, there is a sequence, there is a site where ribosome can bind. Ribosome can bind basically in prokaryote, it is a signal sequence, as you all know. So at this sequence, at this sequence ribosome bind, both subunits of ribosome bind together, and that leads to the formation of another product, which is any protein, right? So basically these two subunits when combined together on the mRNA, then these subunits leads to the formation of a product, leads to the formation of a protein product. And what we can do with the help of this regulatory RNA, when these regulatory RNA attach to the site where ribosome can bind, after that it can enhance the expression of the protein formation, or it can inhibit the expression of the protein formation. So basically here RNA act as a regulator. So with the help of diagram you can easily understand what is happening here. So first of all I want to tell you about the definition of regulatory RNA. So here RNA element within an mRNA, as the name indicate, like it is an mRNA, and at the site in the mRNA, like it is a site where any RNA element, this RNA element that switch between two conformation upon exposure to a small molecule or ligand, like there is a molecule or any stimulus or any kind of ion, or any kind of particle which can attach to this site. And after the attachment of this stimulus or this ligand to this site, the expression of this site can be changed. It can enhance or it can inhibit. Okay, so same things are written here, that RNA element, this is an RNA element within an mRNA that switch between two conformation, and upon exposure to a small molecule or ligand or other stimulus, or it can inhibit or promotes the gene expression of the level of transcription, translation, or RNA splicing. RNA splicing is basically performed in the eukaryotes, but in the prokaryote it is not happening. So in prokaryote there is a signal sequence, which is the site where ribosome can bind. So basically this is a central dogma process, like DNA leads to the formation of mRNA with the help of transcription, and after the mRNA there is a protein product formation with the help of translation. So at this site, at this position, we can even enhance, or we can even inhibit the expression of the gene. So here this is known as ribo switch, as the name indicates, switch can on or off the expression of any gene. So ribosome or ribosome binding site can switch on or switch off the expression of the gene. It occurs in the prokaryote cell, but in fungi or plants it can also find. So basically from here the video is start, that as you can see, any mRNA have 2 prime, 5 prime or 3 prime. This is mRNA structure. And on the 5 prime end or on the 3 prime end, there are two type of region that are known as untranslated region. So these untranslated region, now 5 prime UTR, that is 5 prime untranslated region, consist of a specific gene which are responsible for regulation of regulation of mRNA expression. Basically why are they said like this? that at the 5 prime site you all know that there is a promoter, then there is a operator or a regulator, and then there is a gene which can translate into protein. At the promoter site, RNA polymerase bind, and at the promoter site, at the regulator site, ribosome binding site are present at 5 prime, and these regulator these riboswitch sorry ribosome binding site are present at the 5 prime, and at 5 prime and when ribosome binding site are present, ribosome comes and bind here. Both units subunits of ribosome bind here and leads to the formation of a product, that is protein, right? So in the prokaryote there is a signed Dalgarno sequence which indicate the ribosome binding site. Signed Dalgarno sequence at the 5 prime end, it is present here, and here ribosome can bind. When ribosome binds here you can see that when both units of ribosomes are bind here, then it leads to the formation of a protein. Suppose it is a protein A which is formed as a product after the binding of both subunits of ribosome. It is, if it is in a huge amount, like there is a feedback loop. When protein A becomes in like protein A production is in very high amount, then it leads to act as an inhibitor, which is known as a feedback mechanism. So if it is in huge amount, so it will act as a ligand. So now it act as a ligand or act as a stimulus. As I told you that when any stimulus or any ligand bind to the riboswitch, bind to the site at 5 prime and where ribosome bind, ribosome subunits bind, then it leads to the formation of then it leads to the changes in the expression of the gene, right? So when protein A formed in large amount, in very high amount, then it act as an inhibitor or it can act as a ligand, right? So now when we zoom in 5 prime and then we come to know that there are basically two site, two domains are present before the ribosome binding site. Like here you can see, yeah, here you can see this is ribosome binding site, and before the ribosome binding site there are two domains, aptamer domain and expression domain. Ribosome binding site is a site where ribosome subunits can attach, but its binding depends upon the expression of these two sites, aptamer and expression domain. So when ligand is not bound, like when protein A is not formed in huge amount, then there is a no ligand and there is no bounding of ligand with the aptamer or expression domain. So what happens? It leads to the formation of common product protein, right? But when protein A formed in huge amount, in large amount, then it acts as an inhibitor, it acts as a ligand. So this ligand, which is like it is a protein A, it is protein A, and this act as a ligand, and when this ligand bind to the aptamer site, as I told you that at a 5 prime and before the ribosome binding site there are two domain, aptamer domain or expression domain. So at aptamer domain, when ligand bind or protein A bind, then it leads to the changes in the structure of the RNA. So here what happens, it leads to the formation of a hair loop, and due to the formation of hair loop, ribosome binding site which is present here, which is open here, now it hide. This binding site is now hide, so this site is not available for the ribosome. So no ribosomal subunits bind here, no product will form, ribosome has been moved due to the hair loop formation, and this site is now hide, so not available for ribosome. So no signed Dalgarno sequence is available for any ribosomal subunit, and no ribosomal subunits will bind there, and no product will form, no protein A will form. That this is known as feedback mechanism. But this feedback mechanism is controlled with the help of these two domain, that is aptamer domain and with the help of another expression domain. And basically you can see when ligand attached to the aptamer, then there is a changes in the expression domain, and due to the changes in the expression domain there is a formation of a hair loop, and ribosome binding site now hide here, it is not available now for the ribosomal subunit attachment. So this is basically happened during the inhibition of the gene expression when protein A is present in huge amount. So here you can see aptamer domain and expression domain. Aptamer domain is only bind with the ligand. Here you can see that ligand is attached only with aptamer, and expression domain changes after binding with ligand to domain. As the ligand domain with the aptamer, expression domain changes its expression, and mRNA structure gets converted into another form. So here Ribosome will not able to bind with mRNA and not protein synthesis, so ligand blocked the ribosome binding site and gene expression is switched off. As I told you that either regulatory RNA can switch on or switch off the expression. So in this example, switching off the product, switching off the gene is represented here. The ligand can be of any type, like it can be which binds to the aptamer site. It can be of any ion like Mg2 positive, any enzyme cofactor, FMN, amino acids, short nucleic acid sequence like cyclic di-AMP. So this can be act as a ligand. So here I told you that 5' UTR is consist of a specific gene which is responsible for regulation of gene or regulation of mRNA expression. So basically this is the regulatory RNA site, because at the 5' two domains are present, aptamer domain or expression domain. So these are the specific site where we can say that gene expression can be controlled with the help of mRNA. Another example is here you can see the method, second method 2, where you can control the expression of any gene. Like as you can see at the 5' there is a aptamer, there is another degradosome is basically have enzyme that degrade mRNA. So here enzymes are present which can degrade the mRNA, but it remains hide in loop, so expression remains on. Basically degradosome are present in the loop form, so that's why expression domain are completely available, and at aptamer and expression site have no, at aptamer site there is no ligand, so expression domain is available and expression will not change. Signed Dalgarno sequence is available for the ribosomal subunit attachment. But when ligand attached to aptamer, then this degradosome convert into another circle which open, like degradosome is now present in the loop form, it opens now, and when degradosome opens now any degradosome can bind here, and due to which expression domain change, due to which there is a change in expression of the expression domain, and now Shine-Dalgarno sequence is not available for the binding of ribosome. So degradosome now available, now open, so it leads to the degradation of the mRNA. So now RNase E enzyme binds to it and cleave or degrade the mRNA, so no protein synthesis. So basically it also acts as an inhibitor which can inhibit the synthesis of any protein. So it leads to there is a aptamer and expression, but between the aptamer and expression domain there is a degradosome which in normal conformation present in the inactive form, but when ligand attached to aptamer then it becomes active, and now it can degrade the mRNA with the help of RNase E enzyme, and leads to the inhibition of a protein. So basically it was all about the regulatory RNA. I hope it will help you to understand about the regulatory RNA, that how can RNA element can regulate the expression of the gene and can leads to the formation of enhanced product or inhibited product. So basically there are two methods. First method is like direct ligand attachment to the aptamer, and there is a direct change in the expression domain, and there is a formation of loop, and ribosome binding site hide there, that's why ribosome will not attach there and no protein product formation. And in the second method there is a degradosome presence between the aptamer and the expression domain. As degradosome become active, then it leads to the changes in the expression domain. Ribosome will not attach to the Shine-Dalgarno sequence, and degradosome are now active, and RNA with the help of RNase E enzyme, it leads to the degradation of the mRNA, so no protein product will form. I hope this video will help you. Thank you friends.